to the fascinating world of fish health and disease. Today we will dive in deep into the essential technique of fish dissection, observation, and preservation. Fish dissection provides direct insight into the health status of fish. By examining internal organs, we can detect signs of disease, parasite, and other health issues that are not visible internally. Observation of this condition help me in diagnosing disease early, which is crucial for effective treatment and management. Understanding the epidemiology of fish disease, which aids in preventing outbreaks. Conducting research that contributes to the development of new treatment and management practice. Our specimen is Sardinella melanura, chosen for its representative anatomy. Before any incision, we observe the external feature, skin, scale, fins, and gill. This observation can provide initial clue about the fish health status. So we have three objectives: to learn the technique and procedure for dissecting fish, to study internal structure. Second, to identify and understand the normal and pathological condition of fish organ and tissues. Third, to acquire skill in preserving fish specimen for future study and reference.
In my part, I'm gonna talk about the materials and apparatus that we use in this lab of dissections and observation, also preservation of the fish. Firstly, we prepare three fish. Also, we also need dissection kit, gloves, like microscope to observe our cells under the microscope. We also use tissues, glass slide, with cover slip. We also prepare a dissection tray to put all our fish on the tray. We also use ethanol 70% to kill the bacteria, and we also use distilled water, sample bottles, dropper, and lastly alcohol vanillin.
Fish dissection. Fish was put in a dissecting tray. External feature of fish was observed and abnormality was recorded. Sample was collected for further investigation under the microscope. Fish was disinfected using 70% ethanol. Fish was dissected to expose body cavity. Fish internal organ was observed and organs were isolated. Wet mount slide technique. Tray was disinfected using 70% ethanol. Next, blade and forceps was soaked into 99% ethanol. Blade and forceps was flamed and allowed to cool. The gills, fin, and skin were cut. The piece of tissue was put on the water. Drop of water was added on glass slide. Glass slide was laid on tray. Cover slip was laid on top of the tissue. Cover slip was pressed and bubble was avoided. Histology. Fish was clean. Dissecting kit was clean using ethanol 70%. The desired organ was dissected carefully. The tissue was transferred into buffered formalin 10%. The tissue was transferred into boiling solution after 24 hours. The tissue was kept in room temperature. For molecular work, fish was cleaned. The dissecting set was cleaned using ethanol 70%. The dissecting set was soaked in ethanol 99%. The dissecting set was flamed using burner. The dissecting set was allowed to cool at room temperature. The desired organ was dissected carefully. The tissue was transferred into ethanol 70%. The tissue was kept in fridge or freezer.
This is our results for the lab 1, which is we can see here the two of the picture. The tissue cell is in normal or in a good condition, and we don't find any abnormality. The discussion, we observe the fish first, the organ of the fish, and then we found there is no abnormality because the organ cell is normal, and we observe it under the microscope so that we can identify whether the organ is good or not, and all the organ is good condition also.
Conclusion about our report is the microscopic examination of the fish organ confirms that they are in the optimal health. So all organ and cell appear normal with no sign of abnormalities and disease or damage. So this suggests that the fish are well maintained and healthy.
As for introduction of this streak flag technique and also gram staining techniques, streak flag methods are techniques and gram staining are a fundamental techniques that was used in microbiology that play a crucial roles in the identification and characterizations of bacteria. Just firstly, let's look about the purpose of streak flag methods, which is it was introduced by Robert Koch in the late 19th centuries. It is a technique that used to isolate the single bacterial colonies on the agar flag. This method involves streaking a sample containing bacteria across the surface of the agar flag in a systematic manner. This promoting the growth of individual colonies. By isolating single colonies, the microbiologist can study the characteristic morphology and the growth pattern of specific bacterial species. On the other hand, gram staining. It was developed by Hans Christian Gram in 1884, which is it's a technique or which is very different from the streak flag method, which is divide the bacteria into two groups based on their cell wall compositions. This method involves applying a series of dyes to the bacterial cells, which where the gram-positive bacteria retain a purple color due to their thick peptidoglycan cell walls, while for the gram-negative bacteria, it's, it's appear pink or red due to their thinner peptidoglycan layer and the outer lipopolysaccharide membrane. Both these streak flag methods and the gram staining are one of essential tools in microbiology for bacterial identification and classification. These streak flag methods help in isolating pure cultures for further analysis, while as for gram staining, help to distinguishing between different types of bacteria based on their cell wall characteristics. Next is the objectives of our lab is firstly it's to identify the purpose of both the streak flag technique and the gram staining. Secondly, each one the objective is to learn how to do the streaking flag technique and gram staining successfully. The third objective is to determine the difference in the streak flag and gram staining techniques. The last but not least of our objective is to observe and identify the growth of the bacterial colonies on the agar flag.
Hello everyone, my name is MICCLARRY JUGAH. So today I will present about the material we use in lab. First is glove, number 2 marker, number 3 tissue, number 4 ethanol 70%, number 5 is Bunset banner, number 6 is water, 5 bottle, and 7 is light microscope. We use the in lab is 5 microscope, and 8 is glass slide, 9 is bacteria culture, 10 media agar, 11 is indication loop, 12 is a gram stain region, and the lastly is parafilm tape. So this is all the material we use in the lab.
Strict plate method. An inoculating loop was sterile through a flame until it was red hot and allowed to cool. The cooled sterile loop was dipped into sample. Streak was done in a zigzag pattern on the agar. Repeat step 1 to 4 and rotate the agar 90 degrees. Plate was covered and was incubated upside down for 24 hours. After incubation, growth of colony were observed for identification. Gram staining. Bacterial culture was introduced onto the slide using a sterile inoculating loop. 2. Smear was allowed to dry. The slide was passed through heat for heat fixation. Few drops of crystal violet stain was added, and after 1 minute excess was rinsed and dried. Next, few drops of iodine was added, and after 1 minute slide was rinsed and dried. Next, few drops of alcohol was added, and after 10 to 20 seconds, slide was rinsed immediately. Slide was left to dry. 8. Slide was observed under microscope.
This is our results, and as we can see here, both picture or the results here shows that the gram positive here because it shows the purple color. For the discussion, the result shows that the gram positive bacteria appear in the purple color. So why this thing is happened? Because the peptido-like glycan membrane can hold the dye, and when the stain is combined with bacteria in a sample, so the bacteria will either stay purple or turn pink or red. And if the bacteria stays in the purple one, so that means the gram is positive. And if the bacteria turns pink or red, that means the gram will turn into the negative.
In conclusion, the strict plate method isolates bacterial pathogen from fish sample crucial for accurate identification and analysis. It ensures pure culture by separating individual colony on agar plates. Gram staining differentiates bacteria based on cell wall properties. Gram-positive bacteria appear purple while gram-negative purple stain pink, aiding in identifying and treating infection. This technique enables us to observe bacterial growth and classify pathogens effectively. They are essential for diagnosing and managing fish disease properly. In summary, the strict plate method and gram staining are indispensable tools for safeguarding fish health. They provide critical insight for effective disease management and treatment.